Doctora Helen Piwnica-Worms completó su licenciatura en biología en la Universidad St. Olaf. Posteriormente obtuvo su doctorado en microbiología e inmunología de la Universidad de Duke. La doctora se ha especializado en el estudio de la resistencia en cáncer de mama triple negativo. Hoy en día ha publicado 126 artículos, de los cuales 119 son resultado de investigación original. Desde 2013, asumió el papel de viceprovost de Ciencias y profesora de Biología del Cáncer en el Centro de Cáncer MD Anderson. Recibámosla con una calurosa bienvenida.
To also personally thank Luis for his kind invitation to be at this meeting and for his gracious hospitality. So today I'd like to describe our journey that brought us to trying to understand and target triple-negative breast cancer. And so I will be discussing how the cell division cycle and how it's regulated, and then how checkpoints, in this case in the form of DNA damage or replication stress, signals to the cell cycle machinery to bring about cell cycle delays or cell death. And then we want to know how cancer cells derail these pathways so that ultimately we can take this knowledge back and impact patient care. And then I'm going to describe some work that we're doing to really try to understand and target chemoresistance in triple-negative breast cancer. So in Dr. Dimitrovsky's presentation, he talked to you a little bit about the cell division cycle. We know that it's regulated by a family of cyclin-dependent protein kinases. Now, in turn, these kinases are regulated by additional kinases. And two of these we biochemically characterized several years ago, WEE1 and MIT1. They phosphorylate the CDK component to keep the complexes inactive until the right time in the cell division cycle. And when that time occurs, we have a family of protein phosphatases, the CDC25 family of protein phosphatases, and in particular, we've worked on CDC25A. These phosphatases remove these inhibitory phosphorylations and activate the complexes. Now, when we're trying to understand how, when cells experience stress in the form of replication stress or DNA breaks in the form of single-strand breaks or double-strand breaks, those changes are able to signal to the cell cycle machinery to bring about cell cycle delays. And there's a signal transduction pathway that signals this damage. And a key kinase here is the CHEK1 protein kinase. So what CHEK1 does is to phosphorylate the CDC25A phosphatase and license it so that it can be degraded through the ubiquitin proteasome pathway. Upstream of CHEK1 is another protein kinase, ATR. It phosphorylates CHEK1 to activate it. And this pathway is really important for the S and G2 checkpoints. So this is primarily responsible for arresting cells in the S and G2 stages of the cell division cycle in response to damage. But as we heard from our previous speaker, our genome is experiencing stress continuously, just by the virtue that we live in a world with carcinogens and mutagens, and just, a polymerase can pause. And so these pathways really work all the time through our cell division cycle to monitor the integrity and then pause and allow repairs to happen before moving forward. Another downstream target of ATR is the P53 protein. This is a very important transcriptional regulator. It regulates many genes, but one in particular, P21, is very important for regulating the G1S phase transition. And then ATM, protein kinase, responds to DNA double-strand breaks, feeds into this pathway, and ultimately, P53 activity is required for cells to arrest at the G1S transition. And although it's not required for cells to activate the S and G2 checkpoints, it does reinforce these checkpoints. And so you can really lock down normal cells under these conditions. Now, we've been very lucky over the years. There are many drugs that have been developed to target these pathways. They're in preclinical and clinical testing. You heard Dr. Dimitrovsky talk about CDK2 inhibitors. I'm going to spend a little bit of time on our work with CHEK1 inhibitors. Thank you very much. So, the field, my lab included, spent, over a decade really trying to understand the biochemistry of the cell division cycle and identifying and characterizing these checkpoint proteins. And then we came to the question of how we could ultimately use this information to target cancer cells. So it ends up that replication stress and weakened checkpoints are a universal feature of cancer cells. Most cancer cells derail the p53 pathway, either by deleting it, mutating it, expressing small inhibitory RNAs. And so most cancer cells will not arrest in the G1 phase of the cell cycle in response to damage. And so if you look at this pathway, as I said, tumors inactivate or express mutations of p53, which means that they're unable to arrest in the G1 phase of the cell cycle. So we discovered several years ago that if you add a CHEK1 inhibitor, now you can inhibit all of your checkpoints, and what you observe is preferential killing of p53 deficient mutant tumor cells. And so this became a rationale then for a clinical trial that we initiated when I was at Washington University School of Medicine. Now, at the time, there were no selective inhibitors to CHEK1. We demonstrated that UCN01 was a very potent inhibitor of CHEK1, but it was a very dirty drug at the time. And so our clinical trial, there were 25 patients with metastatic disease. They were given irinotecan, which is a topoisomerase 1 inhibitor, induces single-strand breaks, followed by UCN01 over this course. And 60% of these patients had a clinical response. But what was particularly interesting, 10 breast cancer patients were enrolled on this trial. 4 of those 10 patients had triple-negative breast cancer, and all 4 responded, okay? So that was exciting at the time because, as you know, triple-negative breast cancer is once those patients fail chemotherapy, there's really very little in terms of targeted therapy we can offer them. So we were able to do a few correlative studies at this time. We were able to determine p53 status in 3 of the tumors, 2 from triple negative, 1 ER positive, and correlate with response. So partial responses were observed in 2 patients with triple negative breast cancer. When those tumors were sequenced, one of the tumors had a mutation in p53, the other had very high levels of nuclear p53. And whenever you see that, you know it's mutant p53. Now, the progressive disease was observed in a patient with ER-positive disease. Her p53 the tumor p53 was sequenced and shown to be wild type. So there was a correlation between p53 status and how these tumors responded to this therapy, but it was, we could only do correlative studies in a very few of these patient samples. And so at that point, what I'm quoting Socrates here, the only true wisdom is in knowing you knew nothing. So I'm a basic scientist, I'm a biochemist, and this was really my first foray into really trying to translate our studies. And what I quickly realized is that the patient was a very difficult model organism to work with, and that we needed better ways to, better laboratory, things to do in the laboratory that could help us then understand this disease. And so we started setting up then patient-derived xenograft models from patients with triple-negative breast cancer. And we use a methodology that was first described by Charlotte Cooper-Wasser. And so what we do is we take immune-compromised mice and we humanize their fourth mammary fat pads. And so the way we do this is we take normal human stroma that has been immortalized with telomerase and expresses GFP, and then we engraft that into the mouse. And then when our surgeon colleagues or radiology colleagues call us with samples from the patients, we're able to immediately bring these samples back to the laboratory, process them, and get them into the mice. So we either get biopsy samples or fine needle aspirates. We then dissociate these into organoids, implant them into the humanized mammary fat pads of these mice, and once we, if these tumors take, then we're able to passage these tumors ad infinitum to get as much tumor and as many mice as we need for our studies. So here's then a preclinical study that we set up. We engrafted mice with a tumor from a patient. Again, she had triple-negative breast cancer, and her tumor was wild type for p53. or another cohort of mice with a tumor from a patient whose tumor expressed this gain-of-function mutant, R175H. These mice were then divided into different cohorts where we could treat with vehicle, we could treat with the DNA-damaging agent, which is irinotecan. By this point, there were more selective CHEK1 inhibitors. At that time, we used the lead compound of AstraZeneca, their CHEK inhibitor, and then we did a combination. And what you can see here, this is monitoring the tumor volume over time. On your right, my left, these were mice engrafted with the tumor encoding a wild-type P53, functional P53 protein. And you can see the tumor doesn't respond to any of the treatments. But if you look at the mice engrafted with the mutant P53, you can see that vehicle and the CHEK1 inhibitor have very, very little effect on tumor growth. However, irinotecan has some effect, and then the combination much more. And when you see that actual dip, these tumors actually necrosed and fell off the animal. And we only did 4 cycles of treatment, so we don't know if we had gone longer, if the resistance would have happened. But if you then look at the lifespan of the animals, again, you see animals engrafted with the wild-type P53, there was no effect on lifespan. Mice engrafted with the mutant P53, irinotecan extended the lifespan somewhat, and the combination even more. But now, many of you work with triple-negative breast cancer, and you know it has a lot of intratumoral heterogeneity, and we took tumors from 2 different patients, and we're trying to correlate their response to P53 status. But how can we do that given all the other changes, genomic changes? And so what we did is we went back into the lab and we took the tumor that was wild type for P53 and we knocked it down. And you can see here, we got significant knockdown. In the bottom panel, if we treated the tumors with ionizing radiation, you can see the control cells. We had a nice, robust P53 response as indicated by the accumulation of P21. In the case of the P53 deficient tumors, we greatly attenuated that response. So we then put these cells back into the mice and we treated them again. And so what you can see here is they were treated with either vehicle, the CHEK1 inhibitor, irinotecan alone, or the combination. And you can see nothing that we did really, and we're looking at cleaved caspase 3 here. Really nothing that we did with the wild-type tumors had much of effect, but you can see when you knock down P53 and then subject them to the combination therapy, you get a nice now response and a lot of apoptosis. So this argues that P53 status is a significant predictor of response to therapies that combine certain DNA-damaging agents with CHEK1 inhibitors. Now, there's still a lot that's needed in this space. Certain tumors are very sensitive to single-agent CHEK1. And we need biomarkers that are able to predict that. And so Lauren Byers, who's a really fabulous physician scientist at MD Anderson, recently did a study where she showed certain small cell lung cancers responded very well to single agent CHEK1. And there's a report of B cell lymphomas as well, and this correlated with MYC activation. The response to CHEK inhibitors does not always correlate with P53 loss. So you can combine, for example, CHEK1 with gemcitabine, and you can kill both P53 deficient and proficient tumors. And it's a different mechanism. This is more of an S-phase catastrophe. P53 loss does not equal P53 mutation, so it's going to be important to know if different mutations affect the response of these tumors differentially to these inhibitors. Again, we need biomarkers to predict sensitivity to combination therapies that don't use chemotherapy. There have been certain preclinical studies showing that you can combine CHEK1 with ATR inhibitors or CHEK1 with WEE1 inhibitors. And then finally, we're very interested in the mechanisms of tumor cell killing. So I'm going to move now to work that we initiated when my lab relocated to MD Anderson Cancer Center 4 years ago. And one of the great things about our institution is the large number of samples we can get from our patients and the great collegiality of our medical colleagues there who really want to work with us to tackle these key unmet issues in cancer treatment. So we've now we're generating a couple very exciting PDX collections that are aligned with clinical trials. In the first trial, which is run by Stacy Mulder, We're obtaining serial biopsies from patients with treatment-naive triple-negative breast cancer, both pre- and post-therapy. This is from 360 patients. And at MD Anderson, the first-line treatment is adriamycin and cyclophosphamide. I'll refer to this as AC throughout my talk. Jennifer Litton has a very exciting trial as well. She's treating patients with germline mutations in BRCA1 and BRCA2 frontline with talazoparib. And we're getting biopsies, again, pre- and post-therapy. So some of the unique features of these collections, what we found is we can use fine needle aspirates, and this works very well because they take less processing and we can get them into the mice much more quickly, and so our take rate is very good. And in addition, patients don't mind FNAs, repeated FNAs, as much as they mind repeated biopsies. We are able to generate models from treatment-naive tumors, which is very nice. All of my previous work was done on tumors with, that had progressed after treatment. We have serial tumor samples throughout neoadjuvant treatment, and we can match these to the patient data. So all the PDX models are matched with patient and clinical response information, and we have now a collection of BRCA1, BRCA2 models, both pre- and post-therapy. So what are some of the major questions we're addressing with these models? So we're very interested in identifying, characterizing, and targeting chemo-resistant triple-negative breast cancer. We have a big metastasis program. It ends up when you put these tumors into the mammary fat pad of the mice, they will metastasize and go to the same organs that breast tumors do in humans. So they'll go to brain, they go to bone, they go to liver, they go to lung. And we have a big program in trying to identify mechanisms of resistance to these PARP inhibitors. But for the purposes of my talk today, I'm going to be really focusing on this important problem in triple-negative breast cancer. So it ends up that half of patients with newly diagnosed triple-negative breast cancer respond very well to chemotherapy. So nearly 50% will have a complete pathological response, and you can see on the blue line they do very well. However, it's the patients that do not respond or that have residual tumor burden after therapy that have very poor metastasis-free and overall survival, and that's indicated by the red line and the star. So again, at MD Anderson, our standard neoadjuvant chemotherapy for patients is AC, 4 cycles of AC followed by surgery. And so the way this trial works is patients are enrolled. We get a pre-treatment biopsy that is used to make our models, and we perform RNA sequencing, whole exome sequencing, and histology on those. Then the patients go through 4 cycles of AC. Any patient with residual disease, we get those tumors. Those patients then go on a variety of more targeted protocols. And if at the time of surgery they have residual disease, we also get tumors. And so we have many of these models now that we can begin studying. So here's just a cohort. When we put these models into the mice, then we want to ask, how do they respond to AC? So you can see anything outlined in green. These are our models made from treatment-naive tumors. And you can see they fall into various groups. Some will have a response initially, but if you only do 1 cycle, they will regrow. Others have slowed growth, and still others, if you look in the bottom panel, have no response at all. So if you take one of these tumors, we're going to be calling this PIM1. The black line is vehicle only. You can see the tumor grows. The red line is if we give 1 treatment. So what happens is you debulk about 80% of the tumor, 20% remains, and if you give 1 cycle of treatment, you can see those tumors regrow. That's the red line. The green line is 2 cycles of treatment. So if you now give a second cycle of treatment at the tumor nadir, so at about 21 days, you can see, again, you get this delay, but the tumor will grow. And you can see here out to 4 cycles. You can hold the tumor as long as you're treating with AC, but you just cannot kill these residual tumor cells. And these tumors will metastasize in the mice. So if we Look at the lungs in the mice that have been treated with vehicle only. That's circled or outlined in black. You can see a lung tumor. And even the mice, after 4 cycles of AC, have lung tumors. Now here's if we do a different type of dosing schedule where we let the tumors regrow before we redose. Again, you can see the tumors continued to respond, but it's an attenuated response. And you more quickly generate these resistant tumors that don't respond as well to AC. Now, we had one patient who came in. She had a primary tumor in her breast and a metastatic lesion in her chest wall. And we were able to develop mouse models for both of these. And so, again, the red line is from the primary breast tumor. And you can see after 1 cycle of treatment, again, the tumor, about 80% is killed and then continues to grow at about 40 days. The metastatic tumor, you can see at the onset, has already accumulated more of these AC-resistant cells. And so again, it's enriched for this chemo-resistance phenotype. And here's this metastatic lesion. If we continue to treat it, again, you can see 1 cycle of treatment, you get regrowth. You can go out to 6 and 8 cycles, and again, never kill these tumor cells, but you can hold them at bay. But we know that AC is very toxic to our patients, and so we can't continually treat them with AC. And so we need to come up with ways to really understand this problem. So I'm going to be, we really have gone deep into 3 of these models, and we've chosen 3 models that initially respond to AC, but then regrow. So again, PIM1P is the primary tumor from this patient that presented with metastatic disease. PIM-M is her metastatic lesion, and then PIM5 is an independent model. So what we do is we put these mice on, we treat them with AC, we let the tumors grow to a certain size, we then remove some of the tumor, we then treat with 1 cycle of AC and harvest the tumor when they hit this about 21 days where we've killed the bulk of the tumor, but this 20% is remaining. And then we collect the cells again when they regrow. And we look at them, we look at their histology by H&E staining, we carry out RNA-seq and reverse protein phase arrays, which looks at the proteome. So here's what the tumors look like. I think it's, your eye can quickly see that the pretreated and regrown tumors look pretty similar, but these residual tumor cells look very different. AC treatment induces stromal infiltration, extracellular matrix deposition, and tumor cell pleomorphism. But what regrows, you're going to see, basically recapitulates the pretreated tumor. So if we stain for collagen, you can again see a lot of collagen deposition in this residual state. If we look at phosphohistone H3, which is a marker of DNA double-strand breaks, you can see they're present in the residual tumor, and this is even after 21 days of after that initial AC treatment. Now the cells stain for phosphohistone H3, a certain fraction of these cells, as well as Ki67. So it's not like they're senescent. They are still continuing to proliferate in this AC residual state. Now, if we take the RNA and do a principal component analysis, what you can see, if you look at all the green dots, they cluster. So the residual tumors in each of these models clusters very closely together and away from pretreated and regrown. But you can see the pretreated and regrown cluster together. So we're basically, this residual tumor state is able to regrow and then reform a tumor that's very similar to the original tumor, the pretreated tumor. Here's looking again at the RNA-seq data. Now it's important to know when we do RNA-seq, we're able to subtract out all of the mouse sequences. And so we're only looking at the epithelial tumor cells in this analysis. And if you look at the green dots, that's the residual tumor state, whereas the other dots are the pretreated and regrown. So again, you can see how that residual state has a unique transcriptome compared to the pretreated and regrown. And if you combine all of the RNA-seq data to ask, what is similar between these three models in this residual state, because ultimately what we want to do is understand this residual state so that we can target it and kill those cells. You can see certain pathways, cytoskeleton, cell adhesion, et cetera, that are commonly deregulated in these residual tumors. Now here's the RPPA data. So again, this looks at protein abundance and activation state of these proteins. Now, in this particular data set, we can't separate the mouse stroma from the epithelial tumor cells. So this is going to be a mix. But the important point here is that, if you look at the significant differences between the regrown and pretreated tumors, there's no significant difference in terms of this proteome. And so again, it's telling you that although the residual tumor has a very unique transcriptome and proteome, it's able to recreate the proteome of what we saw in the pretreated tumor. So to summarize this part, the tissue architecture of residual tumors was distinct from that of untreated and regrown tumors. And AC treatment induces stromal infiltration, extracellular matrix deposition, and tumor cell pleomorphism. That the transcriptomes of pretreated and regrown tumors are highly similar. By contrast, residual tumors harbor unique transcriptomes. And while there were numerous significant alterations in protein levels and protein activation states in residual tumors, significant changes were not observed between pretreated and regrown tumors. And so these results suggest that residual tumor cells, or what we call persister cells, enter into a drug-tolerant state that can be reversed once treatment is halted. And these tumor cells, a major fraction of them, again, become sensitive to AC treatment. So then we wanted to ask, is the ability of these persister cells to survive AC due to mutational or non-mutational mechanisms of drug resistance? So we're going to show you, we used barcode-mediated clonal tracking and whole exome sequencing, where we can calculate mutant allele frequencies to distinguish between these two possibilities. And we know from the work, there's been a lot of work by Jeff Engelman in non-small cell lung cancer, where he's shown that there are pre-existing mutations as well as adoptive mechanisms that give rise to drug resistance in the case of targeted therapy, EGFR inhibitors. So these lung cancers will have a minor population that already has a resistance mutation that then can outgrow during treatment. But again, this is targeted therapy, and we're using chemotherapy. And then I'm going to use this PIM1P model for these detailed studies, because as I will show you, it really retains the intratumoral heterogeneity and the characteristics of the original patient tumor from which it was derived. So again, this was a patient that came in. She had a primary breast tumor and a metastatic lesion to her chest wall. So we were able to get her primary tumor and make a PDX model. We got her blood for germline. And this patient, she was an elderly patient who already had metastatic disease. So she was not treated with AC. She went right to paclitaxel. She did not respond, was put on gemcitabine, and quickly succumbed to her disease. And so I'm going to be using this model, and what I'm going to show you is whole exome sequencing and clonal clustering. So this will show you that the intratumoral heterogeneity is largely maintained between the PDX model and the patient's tumor. I'm going to show you the response data of the PDX tumor. It's very similar to how the patient's tumor responded. We're going to look at the whole exome sequence of pretreated, residual, and regrown tumors. So that will allow us to calculate mutant allele frequencies. And then the barcoding data, which allows us to follow the changes in clonal architecture throughout a single cycle of AC treatment. So in the top panel, you're looking at the FNA from the patient sample and then the PDX. So you can see overall the cells look very similar. The best data here is the clonal clustering data. So you've got the patient tumor along the x-axis and the PDX tumor along the y-axis. And you'll note most of the abundance of most of these tumor subclones are maintained in the two samples. Now, you do see some that are enriched in the patient tumor, relative to the PDX tumor, and others that are enriched in the PDX tumor relative to the patient tumor. But overall, this really shows a high level of conservation of intratumoral heterogeneity in this particular model. And then again, these circus plots just show how really messed up these tumors are. So when we treat the PDX, the mice engrafted with this PDX tumor, you can see they don't respond at all to paclitaxel, just as the patient's tumor did not respond to paclitaxel. Gemcitabine, we got responses over 2 cycles and then quick resistance. So again, it's very similar to what we saw with the patient. So we went on to do whole exome sequencing to calculate mutant allele frequencies. And at the end of the day, if you get a result where you see enrichment of these mutant allele frequencies in the residual tumors compared to the pretreated tumors, this suggests that there are mutational mechanisms of resistance, so that some of these subclones have mutations that confer resistance to AC. If you don't see any significant changes in these mutant allele frequencies during a course of AC treatment, then this argues for non-mutational mechanisms of resistance. So this is the data. You can see on the left, really across all the samples, there's no significant difference, okay? So there's no major shifts in any of the mutant allele frequencies. You can see that a lot of mutations are maintained at low frequency, others at higher. But after 1 cycle of treatment, if you compare the residual tumor to the pretreated and then back to what regrows, there's really very little change. And so these results indicate that there are no resistance-conferring mutations that are accounting for this AC resistance. So it's not like with the targeted therapy with EGF receptor where you have these clones that are already resistant. So now the barcoding, what we do for the barcoding is we take, again, these tumors out of the mice, we dissociate them into single cells, and then we transduce them with lentiviruses. And this is a barcoded lentiviral library that has 50 million distinct barcodes. So we're able then to individually mark these tumor cells. We then, they carry a drug marker, so we culture them in 3D cultures for a very short time, just so that the drug works. We then take out some of the cells for our reference. We then implant these in a large cohort of mice, and then remove some at pre-treatment, after a single cycle of AC treatment, and when the tumor regrows. Okay, so, and then we're gonna isolate the DNA, we're gonna do high throughput sequencing of the barcodes. So again, in these experiments, if you see barcode enrichment, then that means clonal selections occurred and that certain subclones, again, have an intrinsic ability to survive AC treatment and then exist in this residual state. If you don't see that barcode enrichment, then it suggests that it's really stochastic, that any cell within this population can ultimately survive AC treatment. And so the barcode, what I'm showing you here are the number of unique dominant barcodes in the reference sample, which is very high. Whenever you go into the animal, you're gonna lose some of these barcodes. But if you compare untreated to the AC residual, you see that the number of unique barcodes are maintained, which means the clonal architecture is maintained. So this result suggests that all subclones have equal probability of surviving AC treatment. However, if you look what grows out, now you can see that only 25% of those barcodes that were in the residual state are able to repopulate to reform the tumor. So that says there's a bottleneck. Any cell can initially respond and sit in this residual state, but only about a quarter can grow to repopulate the tumor. So thus, although all subclones have equal probability of surviving AC treatment, only a subpopulation is capable of repopulating the tumor once treatment is halted. So taken together, these results suggest that there are non-mutational mechanisms of drug resistance that account for this AC resistance in treatment-naive TNBC. And again, this is a single cycle of treatment. And that AC-treated tumors enter a reversible drug-tolerant state, and that a subpopulation of these persister cells is capable of repopulating the tumor with AC-sensitive cells once the treatment is halted. So then we want to know, how do we target this drug-tolerant state? And how do we target non-mutational resistance? And the important question is, do these persister cells have vulnerabilities that we can target? So we have a great assay now. And so what we can do is we can take these cells and we can treat, and at 21 days, we know that we've debulked 80% of the tumor, and we're now enriched for these persister cells. So now we can come in with any agent we want and ask, can we, the goal is to kill these cells, and so how are we going to do that? So as we looked through our transcriptome and proteome data, one of the top pathways that was enriched in these residual tumors was oxidative phosphorylation. And so our Institute of Applied Cancer Science at MD Anderson has made an OXPHOS inhibitor. It's the IAX 10739. And so what we did is we, again, set up a preclinical trial where we implanted the tumors. The blue line is vehicle only. You can see the tumor growing. What is the color? The red is the OXPHOS inhibitor alone. You can see that as a single agent works somewhat to delay tumor growth. The purple line is AC treatment. So again, you see the tumor respond and then begin to regrow. But now if we give the OXPHOS inhibitor at the time, at this 21-day nadir, now you see a significant delay in the ability of these tumors to regrow. Unfortunately, we didn't kill them. They will regrow, but we can then come in with another cycle of AC and they're still resistant or sensitive to AC. So what we're doing now is we have started a very active collaboration with Andrea Califano, and what he's developed is algorithms, which he calls OncoTarget and OncoTreat, and he can take your RNA-seq data and then mine that data to identify drugs already that are in preclinical or clinical testing, and to predict which ones of these will work on your particular tumors. And so just shown here is an example taken from his work where he had a triple-negative breast cancer from a patient that had failed 3 previous lines of therapy. And his OncoTreat analysis identified several agents that would impact this tumor. And as you can see, every single one of them had an impact on these tumors. And so we have a big program now working with IAX, with many of these different models, to, again, try to kill these tumors in the residual state. Because it's quite clear from the work of Jeff Engelman in lung cancer, if you don't get rid of these persister cells, they can sit there and then eventually gain resistance mechanisms to once again grow. And so I think it's really important that we understand this drug-tolerant state, we understand the unique properties of this state, and then use this data, again, to try to kill these tumor cells. And it will be really interesting now to understand if every triple-negative breast cancer will be different, or if there's going to be really uniform properties that are common in every case with these, in these persister cells, so that we're not going to have, an individual strategy for every single patient. We can have, some common things that will attack all triple-negative breast cancer when it enters into this state. Okay, so I'd like to acknowledge the important people in my lab who have done the work. Gloria is circled here. She is a fabulous postdoc who's done all of the work on the chemoresistance in these triple-negative breast cancers. And this is the PDX team. They're fantastic surgeons that, you can imagine when they have 360 different tumors at different stages that they're working with to grow. And obviously, we can't do this work without a village. We have really wonderful collaborators at MD Anderson, within the Institute of Applied Cancer Science, We interact with our breast medical oncologists, our surgeons, our radiologists, our bioinformaticists. It's really a wonderful environment to try to tackle this big problem. So I will stop here, and I thank you for your time. Muchos gracias.